All right. Hey, everyone. Welcome back. We're diving into some pretty wild science today.
Yeah.
Optogenetics,
Yeah, optogenetics.
using light
hmm
to control brain activity.
Exactly.
You guys sent in some incredible research on this. And
Fascinating stuff.
yeah, and it's mind-blowing. How are scientists using light, and then on top of that, gene editing, to actually understand and treat, potentially, brain conditions?
Yeah, it's really remarkable what they're doing these days. I mean, this field of neuroscience is just moving so fast.
So it's like straight out of a movie.
It is.
So first thing that popped out to me, going through these notes, algae genes in mouse brains. I mean, when
Wow.
I first saw that, I was like, wait, what? What is the point of that?
Yeah, I mean, it does sound pretty wild when you first hear it, but it's
Yeah.
actually a really ingenious approach. So, scientists took this gene from algae,
Okay.
and specifically, it's a gene that's sensitive to light, right? It reacts to light. And they inserted it into the brains of mice. And they do this because now that gene allows them to activate those brain cells using light. So it's like
Oh.
installing a tiny little light switch in the brain.
So shine a light, activate a brain cell. That's
Yeah.
wild.
Yeah.
How does that actually work in practice?
So researchers like Carl Deisseroth, for example, at Stanford,
Yeah.
have been doing some really incredible things with this. So they've actually been able to show that by shining different colored lights on these mice, on these modified mice, they can actually influence their sleep-wake cycles.
Really?
They can literally turn their sleep on and off with light.
Hold on. Different colors cause different reactions.
Yeah, so it all comes down to the specific type of light-sensitive protein
Okay.
that the algae gene produces.
Right.
So different versions of this protein are going to respond to different wavelengths of light.
Okay.
So by using a specific color, researchers can basically target and activate only those neurons that contain that corresponding protein.
It's incredible.
Yeah.
But how do they even get that algae gene into the mouse's brain? You can't just like inject it with a syringe, right?
No, it's not quite that simple, but it gets even more interesting here. So the delivery system they actually use is a modified virus.
Wait, a virus?
Yeah.
Like the kind that makes you sick?
Now, before you get too worried, these aren't the kind that are going to make you sick, right?
Okay, good.
Scientists take a harmless virus and they basically re-engineer it.
So they're like microscopic Trojan horses sneaking in the good stuff.
Exactly. That's a great way to put it. So they remove the harmful parts of the virus and they replace them with that desired gene, which in this case is the algae gene. And then they inject this modified virus into a specific region of the mouse's brain.
And then this virus delivers the gene to the brain cells.
Yeah, exactly.
Right.
And then those cells are now producing that light-sensitive protein, and that's what makes them responsive to that light stimulation.
That's incredible. So we've got this like incredible level of control now over brain activity with light. And then reading through this research, it seems like CRISPR is kind of the next step.
Absolutely. Yeah.
So where does that fit into all of this?
So CRISPR, which is a revolutionary gene editing tool, it basically allows us to take this optogenetics to a whole new level of precision.
Oh, so.
So instead of just inserting that algae gene into any neuron,
Right.
CRISPR allows us to target very specific types of brain cells.
So now we're talking about treating maybe very specific brain circuits that are involved in things like Parkinson's.
Exactly, yeah. That's the hope. So Parkinson's disease is characterized by the degeneration of these dopamine-producing neurons in a very particular part of the brain. And with CRISPR, we could theoretically target those specific neurons, insert that algae gene, and then using light, you can stimulate those neurons to produce dopamine and potentially alleviate the symptoms.
So it's like going from using a floodlight to a laser pointer. So much more precise.
Mm-hmm.
But the brain is incredibly complex. How do we know which exact cell types to target with CRISPR for these really complex conditions?
That's the million-dollar question, right?
Yeah.
And it's definitely one of the areas where a lot of the research is really focused right now. So scientists are using all these techniques like single-cell RNA sequencing, and they're basically creating these detailed maps of the brain, right? Identifying the unique molecular signatures of all these different cell types.
Yeah.
And that will really help us understand, which cells are involved in various brain functions and diseases, and therefore which ones to target with CRISPR.
Wow. It's both incredibly exciting and a little bit daunting, to be honest.
Yeah, I agree. I mean, the potential of optogenetics, especially when you combine it with these tools like CRISPR, it's truly remarkable. But we do need to be really careful, with such powerful tools, there's also a lot of ethical considerations here.
For sure.
As we continue to kind of explore these possibilities of controlling the brain with light.
Absolutely, yeah. Big ethical implications there, which we may need to explore in another deep dive.
Yeah, definitely.
But from algae genes and mouse brains to this incredible potential of CRISPR, it's mind-boggling what's happening in neuroscience with optogenetics. So thanks for shining some light, so to speak, on this for us.
Yeah, my pleasure. It's a fascinating topic.
And for all of you listening, we hope this deep dive sparked your curiosity and maybe inspired you to look into this a little more. We'll catch you next time.